Good afternoon, good morning, good evening, everyone. Welcome to today's webinar. My name is Enosh. I'm based here at Azelis in the UK. I will be your moderator and host. I can say that our speaker is online somewhere. So let me just introduce Hamid first before I hand over proceedings to him. Hamid is a senior lecturer at the Department of Pharmaceutics, University of Huddersfield. He holds a PhD in clinical pharmaceutics from University College London. Hamid's expertise is in oral drug delivery, gastrointestinal tract targeting, drug absorption, pharmacokinetics, bioavailability, and bioequivalence. And these are areas where he's published many peer-reviewed and opinion articles. I've personally known Hamid since our days at UCL School of Pharmacy, where we shared the same lab and also a PhD supervisor. I'm delighted that he accepted to speak at this webinar. So today's topic is broadly on the utility of dynamic dissolution testing as a tool to predict GI tract transit and bioavailability of modified release products. Hamid will be speaking for between 30 and 45 minutes, following which we'll have a Q&A session. Please feel free to send in your questions at any point through the chat function, and I'll happily pass them over to Hamid to answer. I'm hoping that there will be a recorded version of the webinar, which will be made available subsequently. This being my very first webinar to host and organize, I welcome your feedback. Any comments on how this can be improved will be very helpful. Also, if you have any specific topics that you would want us to arrange a webinar on, do let me know and we'll be happy to have that organized. So without further ado, I would like now to hand over the proceedings to our speaker, Dr. Hamid Merchant. Thank you so much. Hamid, are you there? You can unmute yourself.
Hi, Enosh. Can you hear me now?
Yeah, I can hear you now. Yes, and I can hear you.
That's brilliant. I couldn't start my video or presentation at the same time you were already broadcasting your video. So it was a bit tricky.
Yes, technology is always tricky, but hopefully we are there now.
I'm still trying to share my screen.
Okay. I believe you can share your screen. Um, do you have a panel on your left? The second, the like two screens, one icons on your left. If you click on there, you'll be able to share your screen with us. If you see menu, yes.
Can you see my screen now?
Yeah, we can see your screen now.
That's brilliant. Can you see this in a full presentation mode?
Absolutely, yes. Yeah.
Brilliant. So thank you very much first, Enosh, for the invitation to speak to in this webinar today. And thank you for everyone to come and attend the webinar. And apologies for the technical glitch to start. This is a different platform than normally we use. And
Yes.
it took me a bit of while to find the right options. So here we are now. So we're going to talk about the dynamic dissolution. Um, we talked about some of the case studies to give you an idea about how to apply the concepts with the dissolution test instrument system, and how to manage the physiologically relevant dissolution testing in a simple laboratory environment. So those who are not aware of where the Huddersfield is from, so that's London. That's around the top. Huddersfield on the north, very close to the cities you might be familiar with: Manchester, Sheffield, Leeds, Bradford. So Huddersfield is just close to Manchester and Leeds and Sheffield, in that triangle round in the middle. So that's where I'm from, University of Huddersfield. And Enosh and I had been together in UCL for a while, a few years ago. So we're going to look at some of the background of where the dissolution testing coming from, physiological relevance to dissolution testing, why it's important, and look at some case studies to see how the concepts can be applied for a bit more predictive outcomes from the in vitro test. So as you all are aware about the range of pH, different enteric polymers available in our boot camp, and different pH thresholds we use, and different backbones of the polymer we have got available, like cellulose base or acrylic base or vinyl based, or some of the natural polymers around. And different pH thresholds mean that we could optimize coatings and delivery systems according to targeted requirements in the GI tract. So particularly today we're referring the applications into oral drug delivery or drug delivery to the gastrointestinal tract. So we focusing on these pH points, which are normally used for targeting drug release into the gastrointestinal tract. I'm sure you are aware of these polymers and then their targets. One thing which is common in all these polymers is the pH trigger, and this is the concept we use for targeting the drug into the different parts of the GI tract, and that has been achieved by a carboxylic acid ionization at a defined pH ionization constant in the dissolution medium, or the intestinal milieu, if you like. And that's the key thing to understand, that how these polymers ionize and how do they dissolve, and how we can then optimize drug release according to our needs for a particular formulations or a product. So one of the old good reference which we always look at is the mechanism of polymer dissolution, going back from dissolution from the bulk polymer mediated by ionization of carboxylic group, and leading these polymer chains being entangled out of the gel layer, coming to the boundary layer, and then diffusing out to the bulk solution. That process is very important to understand in a different environmental settings, where you have got different buffer species present, you've got different buffering capacity, different ionic strength, and that interplay with the polymer is very important to predict the dissolution behavior. Often the models we have in the laboratory for the QC testing are not predictive enough. If you have any change in the formulation, for example, or critical process parameters, which might lead into a critical process change later in the performance of the product, often the dissolution methods we develop for the QC testing are not robust enough to pick up those differences. So IVIVC become difficult, and that can make our life easier if we design methods which could predict the outcomes or the consequences in the in vivo for the little changes in the in vitro which we see. So one of the technique which we looked at personally to understand the behavior of these polymers and their dissolution is the zeta potential. Zeta potential is an interesting technique which can provide useful information about how these polymers ionize in different environments and how they pick up the charge. And as they get ionized, they get a positive charge, which can be then picked up by the zeta potential instrument. And it's normally zeta potential, the Malvern series or other instruments are quite standard piece of kit available in most of the pharmaceutical laboratories. So we thought to utilize this and try to study how these polymers do get ionized, and if their surface charge can be linked to their dissolution behavior. And that was quite interesting, that we found a very good correlation of zeta potential with the dissolution pH. And you can see as the polymers start dissolving, it had a very nice sigmoidal zeta profile. And using that zeta profile of these polymers, then you can work out the ionization constant, and that pKa could then help you understand how the pH thresholds are being set up, and that experimentally determined ionization constant would then be more applicable to then understand how the polymer would behave in a more relevant situation in the in vivo setting, for example, rather than just normal stoichiometric based pKa calculation or a pKa just based on a normal titration curve. So we applied the concept to lots of other polymers, and we found a very interesting correlation with pH thresholds with the ionization constant and the zeta profiles. And the zeta profiles, for example, for SP5055 were different to the higher pH threshold polymers like L100 or S100, for example, which is pH 7 based polymer. So this technique is now published and available as an open access article with this link over here if you wanted to try out. We then also applied the concept to some unknown polymers, some novel polymers in drug discovery, which we are trying to work out as in novel enteric polymers, and also some natural polymers, which already been known in pharmaceutics for drug delivery application. For example, pectin, gum arabic, alginates, and chitosan. And the method was quite quite good in translating into natural polymers. The gums were very difficult to work with initially, but we've optimized and the method to test these natural gums and worked out their ionization constant from the zeta curves. And that's very interesting to see how the reverse enteric behavior of the chitosan matches with the E100 polymer, for example, and that could have got invaluable applications in these kind of formulation targets you have for different products and molecules. So if you look at the dissolution in pH 6.8 usual compendial phosphate buffers, we normally see that the cellulose acetylate, for example, the capsule breaks within 10 minutes and a complete disintegration is achieved within 15 minutes. And that's what normally people expect, that within 15 minutes, the enteric coated products do disintegrate and break down, releasing all the encapsulated drug load into the medium. But when we look at the in vivo behavior of these products, we know that it's not quite quick. It takes about an hour and a half in an average for the formulation to release the drug. And that's quite contradictory to what you find in the laboratory using a compendial dissolution testing. So to apply these concepts into the lab, to predict what's going to happen in vivo, could help you identify a problematic batch in the lab. You could control your process quality parameters more easily, batch to batch variability, process change, site transfers, tech transfers, lots of other things in routine quality control could be a lot more useful in terms of predicting the outcomes without doing in vivo trial again, if your dissolution methods or in vitro testings were more predictive. Similarly, if you look at the methacrylate based products, it's the same behavior. It still takes about an hour and a half to achieve disintegration in vivo in human volunteers. And this is all old data, it's all known, and we see this quite across other polymers as well. So you see from 1 hour to 2 hours, depending on how thick the coatings are and what the pH thresholds are, and some inter-individual variability as well. But when we try to look at the behavior of these polymers, so all these, for example, profiles here show polymer coated tablets using all the range of polymers here, for example, L100, HP55 or 50, or vinyl based coatings here, or cellulose acetate phthalate. And then you can see they're quite similar. The 2 hours is in acid, so no drug release in first 2 hours, and then pretty much every polymer comes out in 15 to 20 minutes window in the beginning. You don't see a real discrimination between these polymers, which that they don't behave exactly the same in vivo. Some of these polymers, for example, L100, dissolve quite at the end of small intestine towards the end spectrum of the pH around more than 7. When we look at the composition of the gastrointestinal fluid, we know that it's very rich, and the phosphate buffers, the compendial mediums, are not reflective of the composition in vivo. And when we look at some of the buffers we worked with, we could see that the Hanks buffer, for example, or a Krebs buffer, represents quite closely to the physiological salt balance in the intestinal fluids, for example. And that ionic balance and buffer species and the capacity is very important to interact with the polymer, its ionization, and then subsequent release of the drug from the encapsulated products. So it's not only for the oral products, the same concept applies to all nanoparticulate-based drug delivery systems which are being injected into the body. Those polymeric species then ionize and dissolve and then dissociate away from the injected site into the nearby vasculature. On that diffusion across then determines your sustained release profile and the half-life of your drug release from the encapsulated dosage system. So it's very important to understand the environment where your product is injected in, whether it's muscles or whether it's gastrointestinal tract, and then understand your polymer. How does it dissolve? How does it ionize? How does it interact with the ionic species around in the tissues? And then how you would work out the release rate constant. So when we check the behavior of these polymeric coated products in more physiological condition, you can see there's a longer lag time. It takes an hour and a half to about 3 hours for different polymers to come out. And you can see there's a broad spectrum now. You can discriminate polymers. Some polymers can be used for proximal GI targeting, and some of the higher threshold polymers can be used for colonic targeting, for example. But if you see their behavior in phosphate buffer or the pharmacopoeial standard quality control test, you would see they all are superimposable here, and you won't discriminate them very well. So that's what you need, in your QC lab, in your developmental laboratory, to be able to identify formulations you are making and how different they are in terms of their performance, so you can have a good idea about the lead selection of a formulated product for your clinical trial later, or for a pharmacokinetic or bioavailability assessment of your product. So that's very important, that you have some discriminatory process and some predictive dissolution testing to guide you into further development, even if the product was new. And if you look at this behavior of these polymers, again, you can predict this based on the ionization profile, which we did using zeta profiling, and there's a quite good correlation between polymers with a higher threshold and the polymer with a lower threshold, and you can see their ionization constant is different. This table summarizes um, the calculated pK value from the zeta profile in comparison to the reported ionization constant in several studies, and some pK has been reported by the polymer suppliers, but those measurements are done in a non-realistic environment in a normal pH-based titration curve, for example, or potentiometric determination. What we found with the zeta profiling is that the zeta max and estimated pK has got a good correlation, and the polymers which has a lower dissolution pH threshold, for example HP50 55 with around duodenal bulb pH threshold, or the polymers like HPMC-LF, acetate succinate polymer, you can see their ionization constant is different to the polymers, for example S100 or L100, for example, with a higher estimated pK. So more the ionization constant is, then obviously you need a much higher pH to get the polymer dissolved. So typically it is 2 pH units more than the ionization constant, which would ensure 99% polymer dissolution at that point. So as a reference here, this study is now being published and it's in open access, so you can download a copy using the link there. So when it comes to bicarbonate buffers, lots of people start panicking. It's not easy to use. It's very difficult. It's tedious. It takes a lot of time, and that's all true. I wouldn't sell you a package which is not very easy to use in the lab. So it is difficult, and one of the key issues is that when bicarbonates react with the protons in your dissolution medium, you will come up with this carbonic acid, which then dissociate further into water and carbon dioxide. Carbon dioxide then bubbles out of the system as a gas. And now that makes things complicated. During a dissolution testing, you have a net loss of CO2, which then shifts the balance towards the left-hand side of the equation. You have a net loss of the buffering species. So buffer gets weaker as you go along. When the buffer becomes weaker, it's not been able to control the pH of the dissolution medium, which is obviously a disaster, because then your conditions are not valid and your dissolution test would not give you right results. So what we did was we come up with different methods of stabilizing this process, and not only just stabilizing this, but automating it, so it just becomes easier and requires less manual intervention to control it. So we come up with an instrumentation which could be then coupled with any dissolution apparatus. It's just an add-on to any USB 234 system. And what the system does is it works on a feedback mechanism. So every dissolution vessel has got this feedback probes which pass on the signals to this controller. And this PLC-based controller programmer then operates the solenoid electromagnetic valves. And then you have these 3 different mixtures of gases, which is then been dosed according to the signals into the dissolution medium. And that controls this equation balance to make sure that you maintain the pH to the target you need for running your dissolution test. So that's how the system works. The concept has already been published, and there are references at the end to look at the further details. So this particular graph is just showing how accurate the pH control is compared to how unstable the buffer is, for example. So if you look at the first one, this one is a very, very low pH base medium. So again, bicarbonate based buffer, but at pH 5.5, a very low pH buffer. But you can see the accuracy of pH control is plus minus less than even 0.01. So it's very good. If you look at the pH 6 threshold, so again, the accuracy is about 0.01. So what you could do is you could set your controller to different pH thresholds, and then the system will maintain that threshold. So one advantage of working with bicarbonate buffer system is that you can play with the carbonic acid and the bicarbonate balance, like the Henderson-Hasselbalch equation. You can play with the buffer equation, and then you can program your controller to control the right pH. So the controller will do everything for you. You just set the pH point. It will work out the right balance in the equation and will maintain that pH. So as you can see, the fluctuation goes slightly higher when you go to the higher pH side. So when you go away from the pKa of the buffering system, you get a bit more fluctuation. But even looking at that fluctuation, this is still within 0.01. So pH 6.5, for example, a set point of pH 6.8, which is a common set point for lots of enteric coated products, the pH fluctuation is plus minus 0.01. So if you look at the y-axis error bars, so this is 6.78 and that's 6.8T. So you're working within that tolerance, very, very tiny tolerance, even though the buffer system is very, very unstable. But this instrumentation can then provide you with that accuracy. So same goes for pH 7 target and pH 7.4, which is often used for colonic targeted products or L100 coated products, for example, or S100 coated products, for example. So when we look at the real pH profile of a gastrointestinal tract, this is a capsule endoscopy work where the pH is monitored in a live human volunteer. So you can see the pH in the stomach is very acidic. After gastric emptying, you see a big shift into the alkaline pH, and you see these kind of a dynamic profile. So gastrointestinal tract is not a static environment, it's a dynamic gut, and the pH changes as you go down the GI tract. And what we wanted to do is now simulate this behavior in the dissolution part. So could you have that dynamic behavior in there? And we were able to do that. So in the system, what you could do is you can do some set points, and you can tell the system that what pH points you want, at what time point you want them, and the system can ramp up and down the pH accordingly by again playing with the buffering components and the Henderson-Hasselbalch buffer equation and monitoring that pH point automatically. This particular graph just shows the buffer capacity of the same buffering system at different time points, different pH points. And the point I'm trying to make here is you're not just changing the pH, you are also modifying the buffer capacity. So the buffer capacity of the medium is also dynamic, it's not static. You can modify the pH, you can control it, and you can optimize it to the requirements you have based on the gastrointestinal environment. And the buffer capacity also changes accordingly. So that's one of the profile we generated for a discriminatory dynamic test for lots of coated products. So the products were proximal GI targeted products, so normal enteric product or gastro-resistant product, and we chose a spectrum between 5.6 to 6.8 to test those products. So you can see we got 5.6 to start with, and then within 5 minutes we ramp it up to 5.6 to 6.0, and then after 10 minutes we go further to 6.5 and then 6.8, and we stop there. So that's the test conditions we have, and the results we found for a range of polymer coated products are quite interestingly discriminatory. So you can see all these polymers have a very different dissolution profile, and that's where you run the test in a dynamic mode. So this is not a static dissolution test, this is dynamic test where the pH is being changed from time 0 to time 160 according to this profile. And if you look at the same products in phosphate buffer, you see this kind of superimposable non-discriminatory profile where every product looks very similar. But we know that that doesn't happen in vivo. These products behave differently in vivo, and the same discrimination you can see here on this spectrum here. So in this one, if you look at the in vivo data for L30D55 coated tablets, we know that it's about an hour it takes to get down and disintegrate in the gastrointestinal tract. And we used that as a validation model and looked at our dissolution profile. So the pH of the duodenal bulb from, again, a radiotelemetric capsule is around 5.6 at that point next to the pylorus in the duodenal bulb. So we wanted to simulate that pH into the in vitro settings to see how this translates into the monitoring behavior. And we found that the dissolution profile was very good in agreement with the in vivo disintegration of, say, 66 minutes, which we found um, from this in vivo scintigraphic study. And that's has a very good correlation with the resolution profile we get by just using a normal in vitro resolution test coupled with our dynamic resolution system. So that's one of the good examples we have that you could actually predict the in vivo behavior there. The other application we had was looking at the dual code system. So um, not sure if you are familiar with the dual code system. This was a system which we designed back in UCL um, to look at how we could expedite the release from these coated products in the gastrointestinal tract. So you can see from the previous in scintigraphic study slide that you had a problem with pretty much every polymer, that the lag time is so high. And what we want to do is minimize this lag time. So once the tablet is emptied from the stomach, it breaches the code as soon as possible, kind of thing. So you get a quicker therapeutic effect from these products. So that's where we were working on a dual code system. And my colleague Philippe was looking after these formulation trials in UCL back then, in a few years ago. And what we found that the dual coat product was performing very differently to the other conventional coated products. So for example, this is a conventional single coated product which takes about an hour to disintegrate, compared to a dual coat product which comes out within half an hour, 15 minutes or so. So that was a very good performance achievement. And we had lots of other formulations of dual coat which we optimized, which were around there. So the tool was very good, not only to discriminate a conventional product with our new product, but also it helped with formulation optimization to find the best candidate of dual coat, which we could take to the in vivo evaluation later. And that profiling worked very well. In vivo trial found quite similar results for the dual coat versus single coat. So we had about less than half an hour for dual coat and more than an hour for the single coat system or the conventional coating system. And when the same products, all the products tested in phosphate buffer, you can see here quite superimposable, similar to release profile, which wouldn't happen in reality. So there's a link to the paper here. This work is also been published. If you wanted to have a look at that, the copy is available from IGP website. The other next stage was to find out that after learning all these knowledge about polymers, seeing how they behave, they're learning their ionization behavior, optimizing the resolution conditions, we wanted to get rid of this coating process and see if we could achieve the same behavior from capsules, which does not require any coating, and you built in the enteric properties within the capsule shells. So what we wanted to do is make the process easier, because lots of you who are working in this field, how difficult the coating process is, and control the CTQs within the coating process, the coat uniformity, the thickness uniformity, and the problem with the edges, and the coating becomes cracked in routine large operations and causing lots of QC failures in GMP settings. So we wanted to have a system where the pharmaceutical manufacturer just buy in capsule shells from capsule suppliers, just fill it in. So no need for coating, and that requires specialist capsule shells to be fabricated using enteric polymers. So what we did was we used some toolings to design our own body and caps of the capsules, and then used pH controlled polymers to design these capsules. So this is a real photographic image of the capsules we made, and this is the resolution profile you can see. So enteric capsules from 3 different polymers, HP55, L100, and ASLF, were made. You can see the resolution profile here, and these resolution profile in the dark symbols are bicarbonate buffers, And then the open simples are again phosphate buffers. You see more discriminatory profile with bicarbonate. And these were the coated tablets from the same polymeric system. So you can see the profile of the capsules were very similar to the coated products using the same polymeric system. So we were able to match the dissolution profiles, which means that the capsules can work as good as the coated tablets are. But that is much easier because you can buy in lots of big supply of capsules from the supplier. So you then shift all the QC burden um, to the third party as well. So the pharmaceutical manufacturing process becomes more easier. The QC is controlled at the supplier end. So you just fill it in basically in your routine operation. So this one was again another profile showing the dissolution behavior in phosphate buffer in this inset diagram compared to the bicarbonate. And this one was the L100 capsules, S100 capsules, which we then tested in pH 1.3, followed by 6.8, and then increased the pH to 7.4. And you can see in phosphate buffer, as soon as pH 7.4 is hit, the polymer just comes, the capsule dissolves. It takes a while in the bicarbonate buffer for the polymer to dissolve. But if you look at just the phosphate buffer USP test, it is pretty much similar to what you expect from the S100 coated products like Asacols in the market. So capsules were quite similar in their performance, if you like. Another application which we were looking at is the API itself. So, for example, different salt forms of ibuprofen. So you can look at the lysine formulation, for example. You've got the sodium salt of ibuprofen. You have a normal ibuprofen acid form. And what you could do is you can have a pH 5.6 bicarbonate media, which is very unstable media. But because of this dynamic dissolution instrument, it's possible to control that unstable buffer in a normal dissolution setting. So what do you see here is a profile for the lysine, very fast releasing, very fast dissolving profile compared to sodium salt, which is again better than the ibuprofen acid. So you can even rank order the dissolution behavior of three different salt forms, which we know that they are in these rank order from the previous experience and also from the in vivo experience, that the sodium salt of ibuprofen dissolves faster, absorbs faster, the pharmacokinetic is superior. Same as for the lysine salt, is a lot better in terms of dissolution and quicker absorption, leading to a quicker Cmax in pharmacokinetics as well. And when you test them in a normal phosphate-based buffer system, you don't see that behavior. The dissolution is so fast, and at least these two products are quite comparable. You don't see any benefit. And within half an hour, you get 100%, whereas within half an hour, you are only around, 50% for normal ibuprofen salt, if you like. And this is the pharmacokinetic behavior in human volunteers, and you can see that the sodium salt, for example, how good the PK profile is in terms of the Cmax and the Tmax as well. This one is the Tmax of three different products: Nurofen sodium, Nurofen lysine, and you can see a quite discriminatory behavior here in terms of Cmax as well. Enosh, can you confirm if you can still hear me? It's been a while I've been talking.
We can hear you. I can hear you very clearly, yes.
Okay. Thank you. Right. So for the delayed release and sustained release formulation, what we did was then we had a spectrum of pH again, right from the acidic pH to the colonic environment. So we were ramped up to 7.4, distal small intestine, and then going down to the colonic environment to pH 6.5. And this whole spectrum can be then modeled into the dynamic dissolution instrument, which can then use for testing products which are delayed release or sustained release, and it will go down to the colonic region for the full spectrum. So we looked at some of the products like mesalazine products in the market, and you can see that the red line shows the actual pH in the dissolution medium while you were running out this dynamic dissolution test, and these colored lines represents the dissolution profile of commercial products of mesalazine. Same dose product, three different brands which uses different polymeric coatings on them. So you would expect a discriminatory profile there. So this has also been published, and copies available on the website. Interestingly, the Mazavent, which is also called Lialda in the US, sustained release product, you can see the dissolution profile is a very straight zero order line in phosphate buffer, and use slightly different pattern here in the bicarbonate buffer. So you can see the mean plasma concentration profile from the in vivo as well, that you have got. The Lialda and Esacol products have got similar pharmacokinetic profile, which kind of suggests that you won't see this zero order profile with sustained release matrix from Mazavent in vivo. So it's behave pretty much similar to Esacol product, which is not a sustained release product. And we can predict that behavior in our dynamic dissolution system. You can see that sigmoidal profile there. And more interestingly, this lag time here is something after gastric emptying you don't see at all, in normal compendial dissolution system. Another one was Nurofen sustained release product. So this is a capsule containing sustained release beads for ibuprofen, and a typical profile going to look like that in phosphate buffer. So you see very quick release. And if you test the same product in dynamic dissolution system, starting from gastric pH, going all the way to the small intestine and dropping it to colonic environment, you see very nice sustained release profile as well. So not like Mazavent that bicarbonates are always bad. It doesn't, the zero order profile doesn't work, but it does work. It depends on the formulation, to be honest. And this is one good example that the sustained release beads in a capsule system for Nurofen has got a very good zero order profile, which is also been seen in the bicarbonate based dynamic dissolution system. And if you look at the elimination half-life from the pharmacokinetic profile, so if you look at the PK data, you will see the Nurofen SR has got a very high T-half compared to the standard Nurofen products. It's about 4 times higher, and that's an evidence of the absorption rate constant being slower, and you see the sustained release profile. So the product does work in vivo. Sometimes you see some of the pH-based triggered products do not work in clinical experience. We got plenty of studies where the intact tablets came out in the toilet, and that's mainly because of the pH variability in human volunteers. So not everybody has got the same pH in their small intestine or colonic environment, which means that the pH-based systems which rely too much on a pH trigger then have got these problems with poor reproducibility or subject to very high intersubject variability. So that's another issue which we already know from our clinical experience and from our in vitro experience as well, that we do see big, large error bars, and even by optimizing those conditions. So what we were interested in was to see if we can simulate these individuals, if we can simulate this behavior in our in vitro setup. So what we did was we come up with these 6 different types of human volunteers. So these are virtual human subjects which were designed based on all possible scenarios. And for example, volunteer 1, very typical average human volunteer, good pH transit time, and pH is 7.4 for a good 2.5 hours. So a good average human volunteer is subject 1. Similarly, subject 2, we then played with a transit time, a shorter transit time at pH 7.4. So the person has got a higher pH, but then the pH is only attained for 1 hour. So what if the formulation do not have enough time? How would that behave in that individual? Similarly, in another volunteer, we've reduced this time to only 15 minutes to see the impact on the performance of the system. And volunteer 4, um, pH never got to 7.4. It stays to 6.8. And volunteer 5, the residence time at the 6.8 is only half an hour. And in volunteer 6, the pH is 6.5 or less. So it's very acidic environment. This simulates a patient where, for example, in ulcerative colitis, you have got very acidic uh, colonic mucosa, bleeding, and the pH is not normal and healthy. So this is an active disease. Some of the patients then go into remission state where the gastrointestinal physiology is better, but some disease patients do not have a normal physiology, and pathological conditions then make the environment of the GI tract change, which makes it more difficult for the pH trigger to work in those volunteers. So then we model them into our dynamic dissolution system. So you can see the pH profiles here based on the settings we have, and then we run the dissolution profile. So volunteer 1, as expected, normal profile. In about between 4 to 5 hours, you get the drug release from these colonic targeted products. Volunteer 2, as expected, had a less quicker release from the product because we had only 1 hour at 7.4. So the pH was changed very quickly. So it took a longer time for this volunteer to get the release from the formulation. Volunteer 3 took even longer because then we had only 15 minutes at a pH 7.4, and that made a difference. Volunteer 4, too long, about 16 hours. This person might have got the tablet come out intact in stools as well. Volunteer 5 is around there. Volunteer 6, tablet never disintegrated within 24 hours. So these were interesting behaviors which we can predict now in an in vitro settings to see how different people would behave with the delivery system, and being able to predict these kind of clinical failures, if you like, which we normally see with these kind of pH trigger system. So that's another application of the system. Then this leads into the floral example where we have got the dual trigger, so the pH and the bacteria trigger, to make it a fault safe mechanism, and that was done again at the UCL. This is now going through the phase 3 clinical trials, being licensed to the Tillot's Pharma, and the product is now being launched in Europe and elsewhere for the inflammatory bowel disease. So that goes to the acknowledgement. I'd like to acknowledge lots of colleagues from the UCL who contributed to the development of various technologies presented in these presentations. It's not all my work. It's lots of people who are working around this, which I've used as a case studies to then look at the application of dynamic dissolution systems. Lots of formulations have been worked by different people, but it's interesting to see that how dynamic dissolution concept can be applied in different formulation settings and then used for formulation optimization, candidate selection for the clinical trial, and also as a guide to develop novel products. For example, we have the capsules, duocode system, floral system kind of thing. So thank you very much. I stop here, and then we have some time for answering questions.
Yeah. Thank you so much, Hamid, for such a captivating presentation and the examples you've given. There have been a few questions. Maybe I'll first quickly start with mine. So in terms of the profiles that you showed, the dissolution profiles, yes, I can say that there's quite a bit of discrimination. There's a lot of discrimination with your system. But did you do any similarity factor testing on those profiles? And how does that appear? I know they raise clearly a difference, but did you do any other modeling to demonstrate similarity or differences?
Yes, we did F1 and F2. I didn't show the data on the slides, and there were some key differences picked up a bit by the F1 and F2 as well. The trouble with F1 and F2 system is that the statistics behind F1 and F2 relies on some assumptions. And if you had so many data points, say, for example, after 85% drug release, or you have got many data points with a flat dissolution curve, which you normally see with longer lag time, then the modeling doesn't work. So you have to be very careful in picking your dissolution points. So you might want to pick up, you have to delete, you have to remove the background lag point. So the baseline, and you have to remove the plateau lines, and then you have to pick up very cleverly points on the actual dissolution curve and make sure you do not have any point more than 85% drug release, for example. So if you program F1 and F2 equations accordingly, then you will see very good discriminatory statistic results from there. If you just put in the whole profile, and F1, F2 would be very misleading. So it's just a bit of caution,
Yeah,
statistics there.
absolutely. Then there's another question as well from, sorry, I can't remember where it came from. You'll pardon me over that. Yeah, Farouk wanted to know whether the enteric capsules that you profiled in your study, are these available commercially, or they were just for the purpose of the study?
We developed them in our labs, so they are not yet available commercially, but we are speaking to some capsule manufacturers to see if we can do a tech transfer from a small scale to a larger scale, because the polymers we used are standard enteric polymers. They're not
Yeah.
patented or proprietary to any companies, but the technical know-how in making capsules might be something which some people might find to reproduce. But the concept is published in open access, so the data is all available. If somebody wants to try it out, it's all there. Compositions and rheology and everything is there. But we are trying to work with the capsule supplier and see if we can translate this into a large production scale, and then once it's been done, that can be available to buy routinely, because polymers are exactly the same. It's not a new material, so regulatory pathway will be very easy to get this.
Yeah. Then there doesn't seem to be any other question, but maybe I can add another question from me. Um, has there been any movement by the regulatory agencies on acceptance of bicarbonate buffers as compendial media?
Um, they again have got quite flexible requirements, so they do not want to put bicarbonate as a mandatory requirement, because lots of phosphate-based dissolution methods are validated with IVIVCs. So if you have got, say, for example, 3 different release profiles, which can be matched with a very good IVIVC for an in vitro pharmacokinetic or biopharmaceutics profile in human volunteers, and you can demonstrate that you can you can control your quality of routine manufacturing, then they don't need to enforce a bicarbonate based system for a routine manufacturing. When it comes to designing your own method, which is more discriminatory, they don't mind using any buffering system, as far as you show the application and IVIVC correlation in there. So I don't think regulatory is a problem. They want to keep it more simple for routine QC testing, as far as it's predicting within the design space. But if the method is not predicting a bad product, then regulatory agency will come back to you and say, look, your dissolution method is not discriminatory. So for
Yeah.
example, if you made 3 products, one very slow releasing, one good one, and one very fast one, and then the pharmacokinetic doesn't match with the profile, then your dossier will not be accepted like it used to be accepted in the past. So you will find it difficult.
Absolutely. Um,
So
I also have one. I think this will probably be up because we only have 3 minutes to go. Do you have any experience? This is from Kat. Any experience with polymer coating of API and its impact on dissolution profiles for suspensions?
yes, I think it's a very interesting question. The API coating is a very good example where you have got a polymer along with drug itself on the surface, and you've got two species dissolving and ionizing at the same time. So if your drug product is also a weak acid or a weak base, then you have got a pH base ionization happening at the surface along with the polymer.
Yeah.
And sometimes interaction of the drug with the polymer is such that it improves the solubility profile as well. So solid dispersions, for example, is another case where uh, the acetyl succinate, for example, HPMCAS, makes very good solubility profile of some poorly soluble drugs. So you might get a product into an amorphous state by using a blend of polymer. Some kind of polymers behave better than other polymers depending on the ionization constant and molecular weight of the drug. And we've been doing some studies to link zeta profiling with polymer selection for those kind of applications where you could actually use a more clever polymeric system which
Yeah.
favors the best dissolution profile of your drug. So you're absolutely
Absolutely.
right. It's a very important area to look at, and this knowledge is very important in terms of ionization constant, in terms of polymer selection, in terms of the ionic species around your administrative site, the buffer capacities, and the environment.
Yeah.
Any
Our time is up. Attendants, thank you so much from my side. And thank you, Hamid, as well, for the wonderful and very informative presentation. Continue sending in your questions if you have. Thank you again. Give us feedback. If you have any other requests for such webinars, let me know, and we'll be happy to oblige. Thank you, and have a good rest of the day. Bye-bye, everyone.
Thank you. Thank you, Inash, for the invitation, and thank you everyone for attending the event today.
Yeah, thank you.